Hello, Biochemistry Simplified. Madam Winnie here, introducing carbohydrates as the first biomolecule. After introducing ourselves to how important biochemistry is, let us now dive into understanding biochemistry. So, I'm going to start by looking at the carbohydrates. Basically, I'll start today by doing the monosaccharides only. So carbohydrates, they are synthesized by photosynthesis in plants, and the best example of the carbohydrates which we know are grains, cereal, bread, sugarcane, name it. There are many examples. So they have a composition of carbon, hydrogen, and oxygen. Classification of these carbohydrates, it doesn't change. We have the monosaccharides, the one sugar, the simple sugars, which you can't hydrolyze further. They are simple such that you can't hydrolyze them further. So the best example of the monosaccharides, we have the glucose, the fructose, and the galactose. Then the second class are the disaccharides. From the name, di means two. So it's a condensation product of two monosaccharide units, but then they are linked together by a glycosidic bond. So a glycosidic bond is found in the carbohydrates. The best examples we know are the sucrose, which is always found in sugar, lactose, which is found in milk, and maltose, which is also a component of Guinness. There is another class which I didn't include here, are the oligosaccharides, but then they are now accommodated in the class of the polysaccharides. Polysaccharides, from the name, it is a polymer of those monosaccharide or disaccharide units, but then it is now a length of those monosaccharide or disaccharide units which are still linked by the glycosidic bond. So the common ones, which are known as the homopolysaccharides, are the starch, glycogen, together with cellulose. I'll start on the monosaccharides today. We will do the monosaccharides in this slide. If you look at the monosaccharides are always drawn into projection. It is either a linear projection, which is always known as the Fisher's projection. Whatever shows on the slide here is a linear projection, which is always known as Fisher's projection, or the Haworth or cyclic projection, which we'll look at it later. So we have to understand the linear projection before we can understand the cyclic projection. So for the monosaccharides that we are going to start off with, we define them as simple sugars. It means you can't hydrolyze them further. They're already simple, so you can't break them down further. So the best example is we have glucose, fructose, and galactose. We talk about one sugar or a simple sugar. They are monosaccharides. You can't hydrolyze them further. They're already simple sugars. And then these monosaccharides, we are going to dwell much on them by looking at how naming of a monosaccharide is done. So naming of a monosaccharide is done based on two criteria. One, the functional group, and two, the number of carbon atoms in the main skeleton. Naming of monosaccharides, the first criteria, based on the functional group. Functional group, it is either a monosaccharide is a ketose, or an aldose. So the aldose is the aldehyde group which is always seen in carbon atom number 1. Aldehyde group seen at carbon atom number 1, they are known as aldoses. Then we have the ketoses. At carbon atom number 2, there is a double bond O. So classification or naming of those monosaccharides is based much on the two criteria, the functional group or the number of carbon atoms. For the functional group, the monosaccharide is either an aldose with an aldehyde group at carbon atom number 1 or a ketose with a keto group at carbon atom number 2. So the best example here is glucose for aldose, while fructose for a ketose. But I mentioned that these monosaccharides can be drawn in two forms. It's either a linear form, which is a Fisher's projection, or a cyclic form, which is known as Haworth projection. When you are told to draw L-glucose, D-glucose, L-fructose, D-fructose, L-mannose, D-mannose, then definitely the L and the D will tell you that the teacher expects you to draw your sugar or your monosaccharide in a linear form. I repeat, if you see the teacher asking that draw L-glucose, D-glucose, L-fructose, D-fructose, L-galactose, D-galactose, then you need to draw your monosaccharide in a linear form or Fisher's projection. Let's see how we are going to draw it in a linear form. When you want to draw D-glucose, for example, remember naming of monosaccharide depends on two criteria. 1, number of carbon atoms in the main skeleton. For glucose, it is 6 carbon atoms. So you draw the 6 carbons, 1, 2, 3, 4, 5, and the 6th one. Then the second criteria is based on the functional group. For glucose at carbon atom number 1. At carbon atom number 1, we have a double bond O, then a H this side. At carbon atom number 1, there is an aldehyde group. It has 6 carbon hexose. Carbon atom number 1, double bond O. So at that point, you talk about an aldehyde group. Then, around the carbon, remember it was the chemical reactions taking place. It has a component of carbon, hydrogen, and oxygen. Around the carbon, if you look at atomic number of carbon, it reads atomic number 6. So the configuration goes to 2 to 4. The second shell needs 4 electrons to be stable. So the bonds around the carbon has to balance with the 4. Then you have 6 carbons down the line. Carbon atom number 1, number 2, number 3, number 4, number 5, and then number 6. The 6th carbon atom puts CH2OH. We are drawing D-glucose. When we are drawing D-glucose, you count carbon atom number 1, number 2, number 3. The OH is put to the left, balance with the hydrogen. And when you're putting the OH, let the oxygen be close to the carbon than the hydrogen. At carbon atom number 3, the OH is to the left. For D-glucose, balance with the hydrogen. It is the property of water, an ideal solvent. So when OH is this side, H is to the other side. Then at carbon atom number 2, number 4, and number 5, put the OH to the right, balance with the hydrogen, you have D-glucose. It means for L-glucose, at carbon atom number 1, we still have the aldehyde group. It is still 6 carbon atoms. Carbon atom number 6, CH2H. When you are drawing L-glucose, it is just a mirror image of D. So at carbon atom number 3, the OH which was to the left now comes to the right, balanced with the hydrogen. The OH at carbon atom number 2, number 4, and number 5 which was to the right now comes to the left, balanced with the hydrogen, and that is L-glucose. Still based on the functional group, the second functional group is a ketose. Ketose, the second carbon atom is a keto group. Fructose is the best example here. Fructose is still 6 carbon atoms, 1, 2, 3, 4, 5, and then the 6th one. At carbon atom number 2, there is a double bond O, that is the keto group. Carbon atom number 1 puts CH2OH, carbon atom number 6 puts CH2OH. At carbon atom number 3, we are drawing D-fructose. The OH is to the left, balanced with the hydrogen. So it leaves you the carbon atom number 4, number 5, the OH is to the right, balanced with the hydrogen to the left, you have D-fructose. Naming of monosaccharides, the first criteria, based on the functional group, we have the aldehyde group at carbon atom number 1, the best example is glucose, and then we have ketose, which is fructose being the best example at carbon atom. The second criteria for naming of monosaccharides, number of carbon atoms in the main skeleton. So if you are talking about the 3 carbons, we're talking about the triose, 4 carbons tetrose, 5 carbons pentose, 6 carbons hexose. That's how simple it is. So the hexose, we had the glucose and fructose having 6 carbon atoms, we named them hexoses. So what you can say here is, based on the criteria of naming of monosaccharides, we combine both the functional group and the number of carbon atoms. So this first one, its name is known as glyceraldehyde, but as per now, it has an aldehyde group at carbon atom number 1. It is an aldose. It has 3 carbon atoms down the line. It is a triose, so the name will be combined, and having the functional group and the number of carbon atoms, it is an aldotriose. Its common name is glyceraldehyde. The second example has an aldehyde group at carbon atom number 1, 4, 6 carbon atoms, 1, 2, 3, 4, 5, 6th one. It is an aldehyde, meaning it is an aldose. 6 carbon hexose, it is an aldohexose. Whatever is shown there is D-glucose. Then we have ketose, carbon atom number 2. It is a 6 carbon hexose, so it's known as ketohexose. The functional group and the number of carbon atoms, its name is D-fructose. That is how naming of monosaccharides is done. It is based on 2 criteria: the number of carbon atoms in the main skeleton, as well as we have the functional group. The functional group. So the functional group the functional group functional group here functional group. So based on the functional group and the number of carbon atoms in the main skeleton, functional group. Functional group. So that is a functional group, and in blue is the number of carbon atoms in the main skeleton. So we call it an aldohexose, an aldohexose, an aldotriose. So that is the criteria for naming